Our presentation today is on exogenous AGEs, microbiota, and their role in chronic diseases. Presentation outline, I'll be talking about what are AGEs and their pathogenic mechanism, what is the role of dietary AGEs in humans, both in health and disease states, do AGEs form in intestinal lumen, what is the relationship between AGEs and the microbiota, and then I will point out to some future directions of research. AGEs are heterogeneous group of compounds resulting from the spontaneous non-enzymatic reaction of the carbonyl groups of sugars with the free amino groups in protein. As you can see in this scheme, the free amino groups reacts then with reducing sugar spontaneously in a non-enzymatic reaction, leading to a series of intermediate reactions that eventually culminate in the formation of advanced glycation end products, AGEs. This again to emphasize, in the gray line, you find that reducing sugar reacting with amino acid leads to the formation of reversible ship base, reversible amyloid products, and then the irreversible AGEs such as CML, carboxymethyl and pentosidenine. This is a so-called Brown reaction or Maillard reaction. However, what is clear is that there are many reactions through which AGEs can be formed, and what is even more important, that you can form AGEs even in the absence of sugars. We care about AGEs in medicine because they are products that lead to inflammation and oxidative stress, which at the end are processes that eventually lead to chronic diseases such as diabetes, cardiovascular, and chronic kidney disease. AGE produce biological effects through two general mechanisms. On the one hand, they cross-link proteins and they directly alter their structure and therefore their function. For example, glycation of collagen fibers in the skin and vessel wall changes the characteristic of this protein and their holding tissues, and this produces wrinkling under the skin and a stiffness of arterial walls. On the other hand, AGEs acting through receptors and on-receptor mechanism are able to activate many cell pathways, leading finally to increased oxidative stress and inflammatory cytokine. Several AGE receptors, some pro-inflammatory such as RAGE and TLR4, and some anti-inflammatory such as AGR1. Actually, the binding of AGEs to AGR1 leads to degradation and endocytosis of AGEs, so it acts as a protective mechanism. Now, AGEs are very well known as compounds generated endogenously in diabetic patients as a result of hyperglycemia. However, fewer people are aware that AGEs are naturally present in all foods, and this amount is greatly increased by processing and/or cooking food under high heat. Actually, heating of foods generates AGEs, and the AGE content increases as a function of the type of food, duration and level of heat, moisture, pH, etc. This is very well understood by food scientists. They have learned how to manipulate cooking techniques to get the exact color, aroma, and taste of food they desire. We and others have demonstrated that a portion of AGEs in food will get absorbed and incorporate into the body AGE pool, becoming there indistinguishable from endogenous AGEs. Now, a high dietary AGE intake in mice has produced almost any known chronic disease such as diabetes type 1 and 2, atherosclerotic disease, renal disease, insulin resistant, impaired wound healing, and more importantly, a low AGE diet has prevented all the same conditions. The fact, however, that a low AGE diet prevents disease in mice does not assure that it will do the same in humans. As a physician, my main interest has been to define if dietary AGEs are relevant in humans. For about 2 decades now, we have been addressing this issue in several studies, and I believe we have answered in the affirmative. But because of time constraint, I will just summarize our result with intervention studies. First, let me provide some background information to better understand how these studies were performed. Modern Western diets are rich in AGEs. Cooking with heat Under dry conditions such as grilling, leads to significant formation of AGEs, especially in animal-derived foods, which are also rich in fat. These images depict the AGE-rich food we are talking about. Different cooking methods can significantly affect the AGE content of food. In this example, we took samples of the same weight from chicken, steak, salmon, and thus exposed them to different cooking methods on the same length of time. And clearly the message here is that different cooking methods produce different AGE content. So the modification of the dietary AGE content is not modification of the nutrients that you eat, but rather how do you cook the food. It is a modification in the culinary technique. AGEs are ubiquitous in food and formed during a series of heat-related industrial processes, including processing, sterilization, packaging, and long-term storage of food products. Home cooking, such as broiling, grilling, frying, and reheating of ready-to-eat foods, also increases the production of AGEs. In general, Western-style diet, rich in fats, processed meat, sweet snacks, and refined grains have very high levels of AGEs. For comparison, a Mediterranean-type diet with more plant-based foods, less saturated fats, whole grains, and minimal cooking contain much less AGEs. This leads to what we call a low AGE diet. Essentially, it's the decreased intake of high AGE containing foods based on existing databases in the literature, and that modification of the AGE content has been obtained through culinary techniques modification, temperature, duration, moisture, pH, and surface of cooking. In a seminal study published in 2002, a group of diabetic patients were randomized to either a high or low AGE diet. Their meals were prepared in the Clinical Research Center, and participants picked them twice a week. And essentially, the results are expressed in the next slide. We present the result here as a percent of change from baseline values because of the different units. First, if you concentrate in the upper panel, you can see that a low AGE diet was associated with decreasing blood levels of at least 3 markers of AGEs, while a high AGE diet produced exactly the opposite. Please realize that levels of blood AGEs were decreased by as much as 40% despite same degree of diabetic control. Up until this moment, serum AGE levels in diabetic patients were thought to result exclusively from hyperglycemia, and therefore this striking fall in levels while maintaining blood glucose control about the same was unexpected and a very novel finding. If you now pay attention to the lower panel, you will realize that a low AGE diet not only decreases circulating AGE levels, but also decreases circulating levels of markers of inflammation such as CRP, TNF-alpha, and endothelial dysfunctions such as VCAM1. The fact that the high AGE diet did exactly the opposite strengthened even more the findings. All these findings have helped us to delineate a new paradigm, the AGE story or hypothesis as we call it, in which circulating AGEs originate not only endogenously but also exogenously from food. Whatever the source of AGEs, they lead to an increased AGE pool with increased oxidative stress and inflammation, which in turn leads to chronic disease. To further strengthen our postulation, we took another group of type 2 diabetic patients who were naturally consuming a high AGE diet and randomized them to either continue the same or go on a low AGE diet for the next 3 months. Patients prepare meals at home under the constant phone and or face-to-face guidance of their research dietitian. Results are shown here only for the low AGE diet and as a percent of change. And again, serum CML came down, markers of inflammation such as TNF-alpha and RAGE came down, markers of oxidative stress such as isoprostane came down, and of interest, HOMA index, a marker of insulin resistance, decreased on the low AGE. This above result, and later what we demonstrated in patients with metabolic syndrome, has supported the idea that the relationship between AGEs and diabetes is not only that hyperglycemia can lead to high levels of AGE, but indeed we are postulating that dietary AGE can by itself increase insulin resistance and therefore induce diabetes the same as in mice. This table shows many other clinical trials with a low AGE diet showing similar results. So nowadays, most people accept that the sources of AGEs are either endogenous and exogenous. And exogenous will include smoking tobacco, eating AGE-rich food, eating fructose-rich food that can lead secondary to increased formation of AGEs after glucose is absorbed into the body, or even the intraluminal formation of AGEs. This investigator then studied the formation of Maillard products during digestive process of meal-resembling system, and essentially they demonstrated the formation of non-fluorescent AGEs during digestion of sugar-containing average meals. The postulation here is that this formation of Maillard product reaction may be possible in conditions where sugar absorption is delayed, such as fructose malabsorption or intake of a fatty meal, and this cartoon again illustrate the same. Now, a percent of the AGEs ingested or formed intraluminal will be absorbed and incorporated into the body AGE pool, contributing to the oxidative and inflammatory state. The remainder will be delivered to the colon, where they could be excreted in the stool, interact with a RAGE receptor in the intestinal wall, and initiate a localized inflammatory process that could become systemic, or actually they will interact with the microbiota, producing either modification of the microbiota, they could be broken down by the bacteria, or can increase the local formation of AGEs. There are many relationships between AGEs and bacteria in the colon. A few examples of clear relationships that can be documented include the fact that some lactobacilli make glyoxylase, the enzyme that breaks down methyl glyoxal and potentially then metabolize AGEs from the diet. In fact, it has been shown the carboxymethyl lysine is degraded by E. coli by several work, and E. coli can actually secrete AGEs and may contribute to bacterial-induced inflammation. This group, this is a study done by Dr. Mastrocola, a group in Italy, looking at mice who were allocated to either control or high AGE-rich diet for 22 weeks, and then the ileum samples from these mice show marked increase in the presence of CML and RAGE in the intestinal wall of the high AGE animals. The work by Dr. Cristina Delgado Andrade in Spain has shown the effect of the high AGE diet on the right microbiome. In this case, rats were fed either a control or high AGE diet for 88 days, and you can see that this has a tremendous impact on the microbiota of these animals. Then the same group led the study to a group of human adolescents on different diets with AGEs, and this again clearly changed the microbiota. Later, a study by Dr. Jakub took a group of end-stage renal disease patients on peritoneal dialysis, randomized to either high or low AGE diet for 1 month, and again, they show effect on the microbiota. So by now, animal studies and human studies have shown the fact that the high AGE diet modifies the bacterial flora. The metabolism of AGEs is summarized here, showing that exogenous AGEs ingested via cooked food or those formed inside in the small intestine can have a partial AGE absorption in the small intestine into the circulation, from where they can be excreted in the urine by the kidneys, can have protein cross-linking directly, or can have cellular effects leading to inflammation and oxidative stress. While the AGE is not absorbed in the small intestine, moving to the large intestine, can either be excreted in the stool or react with RAGE receptors on the surface or react with the microbiota. And again, this is another way to present the same summary. Those AGEs delivered into the colon can affect the microbiota, local fermentation, production of new compounds, including new AGEs, increased colonic wall permeability, leading to local inflammation and inflammatory bowel disease, generating food allergies, and at the same time, this may become systemic inflammation. Let's not forget that tobacco or air contamination AGEs can also be absorbed via the respiratory tract, leading to local inflammation in the bronchial tree like asthma, and potentially absorption to lead to systemic inflammation and oxidative stress. So all of these reflects the AGE hypothesis that, as I mentioned before, state that AGEs coming from endogenous or exogenous sources will increase the body AGE pool, leading to oxidative stress, inflammation, cross-linking, that eventually lead to chronic, non-infectious diseases. What we need to do to strengthen this area is, number one, refine our understanding of the mechanism linking high dietary AGEs to disease by utilizing newer and more sophisticated techniques available already in some other areas. We need to reach an agreement in terms of most representative AGE, the best method to measure it so we can start developing actual guidelines. And we need to refine the interaction between AGEs and the microbiome and identify potential areas of intervention there. Let me just mention the acknowledgement to people who have helped me in all this work, and thank you very much for your attention.